It seemed to be.
You're cleaning up. We're dying. That's what.
You can get in the subway chair.
In your armpit. I older kids that do that.
Don't forget y'all's bag, Armstrong's. Hey, Darcy. Darcy, don't forget that bag with your stuff in it. There you go.
Why did you get the bag?
Because he's Lewis.
You don't need to do anything.
He's like, oh, you have food. I'm here.
Wait, we have to enter them all?
What now?
Don't have to enter all the questions?
Just do your best. Get done what you can get done and you're fine.
Say Anthony, you're all good. If you want to go, if you want to.
We do have a lesson. Oh, we have a lesson.
We have lessons?
We have lesson. I have. Wait, you have lessons. Nice.
I have more in.
Everybody's class today.
Yeah, we got about 3 people.
You know I eat stuff?
Oh, 4 people. Yes, I do.
Cool.
His mother and sister and dad are in Arizona.
Oh, yeah.
Yeah.
Just big chilling.
And he still doesn't have a license, so
Yeah.
I'm gonna keep doing that. I'm gonna pass around these 2 little thank you cards. If everybody would just please sign their name to them and pass them back up to me. These are for people who donated some lab supplies to us for Christmas that have nothing to do with our program. They're not connected to us in any way other than they're nice people. So, don't mind. Thank you. And then we'll get started. I have Sydney and Savannah here, I believe. Yeah. Okay, and Shannon should be in soon.
Hi, Sydney and Savannah.
Mm-hmm. All right, so I twisted.
Dog.
Drew did the same thing.
I have one that twist, but the bottom part twist.
Yeah. Okay, so here we go. We're gonna start on protists and fungi. This is one of those words too. You can do fungi, fungi. People do it all kinds of ways, whatever. I see. This is spoiled brat. So what we're going to be doing here is we're going to be starting from the most.
Anthony, you left your binder.
Anthony, you still here?
Anthony.
No, fungi. Is he still here? He's still here. Okay, so we're going to be starting with our most basic life forms and moving towards our most complex life forms over the next 2 quarters. What's the most complex life form we've got?
Humans.
Had to be right. So we're going to start with these very, very basic protists, and then we're going to kind of move through the layers of evolutionary complexity until we get to our most complex, which is us. Our last quarter is going to be pretty much all human biology, human anatomy, human body systems. Crazy fish just jumped. So we're starting now with the very, very basics. Do you have it? Is he good? I thought I heard his voice. Why didn't he come get? Okay, well, he probably wants to come get that before he forgets it though. So let's look at our homework. In our homework, we discussed the mad cow disease mystery, and we had a little of this discussion in our Zoom class last week. And we also talked about scrapie and kuru. So in 1986, something strange happened to cattle in Great Britain. Some were acting strangely, losing control of their movements, and eventually dying. Farmers watched helplessly as the disease they called mad cow spread through their cattle. The disease affected more than 30,000 cattle in 1991. British scientists carefully researched the veterinary histories of 169 sick cattle, and all 169 had been given feed enriched with meat and bone meal protein from slaughtered cattle. Could this practice spread the disease? Let's think for a minute. Do cows naturally eat cows? No, I mean, you would think, And prior to them unnaturally eating cows, they were also giving them bone meal and parts of sheep, okay? And sheep can pass on a prion that causes a disease called scrapie, which is very similar to mad cow. The meat and bone meal substances added to the cattle feed in Britain were sterilized at high temperatures in excess of 100 degrees during processing. How could any infectious agent withstand that? When scientific researchers studied the brains of cattle killed by mad cow disease, they found that large areas of the animal's brains had been destroyed. Under the microscope, holes, why am I vibrating? Mine, it's okay. Holes in the tissue made the brain resemble a sponge. And because of this, they called it bovine spongiform encephalopathy, or BSC. But the cause was still a mystery. So you guys were given an article about it and you had some questions to answer. So where and when, do you remember at least when, if not where?
Great Britain.
It was going to be 1986.
What places stop exporting?
Well, not on that question yet. How many BSE infected cows have been found to date in the United States?
4.
Three.
It listed 3, but it said there were 3, but then it listed 4. It said there were 3 found, and there was 1 in Alabama. 2 in
Yeah.
Washington State,
And if
it
you
was
Google it to get your most accurate or most current, yep. So since 2003, there have been 6 cases of BSE identified in the US. So 2 in Alabama, 1 in California, 1 in Florida, 1 in Texas, and 1 in Washington. So which countries stopped importing beef from the US after our first incident of mad cow?
Mexico.
Yeah. So USDA's 2003 BSE announcement, most countries banned, all or some, and these included Japan, South Korea, Mexico, Canada, which together had purchased 90% of our beef. So that caused some issues. So how much does Creekstone, the company that was talked about in the little article, contend it lost in revenue each day from the drop in exports?
200,000.
Yeah, 200,000 per day. And infer what the key arguments on each side are. Why does Creekstone think it should be able to conduct its own BSE testing? And why does the USDA oppose that idea?
Creekstone, but it was to be able to continue selling and know that it wasn't
It
infected.
was
To put confidence in the buyer.
There you go. Yeah, basically it. So yeah, I didn't write one for that because it's kind of your opinion. So now we're going to go into some Protist classification. This is a gorgeous diatom. I love diatoms. They're so pretty and they're all different looking and they have these gorgeous like silica shells, basically glass. I mean, they're basically little glass organisms. They're really cool. So our first eukaryotes are going to be our protists. So what's a prokaryote then? First eukaryote, what's a eukaryote going to be versus a prokaryote again? Yeah, yes, nuclear bound membrane. So a protist is any unicellular eukaryotic organism that is not a plant, animal, or a fungus. Now there can be some multicellular ones. Yes.
You said nuclear bound
membrane bound nucleus. Sorry. Yeah, I spoke backwards. So there are some multicellular protists, and they are the fungus-like ones. And what tends to happen with some of them is they all kind of mash together in a colony, and then they sort of lose their individual cell membranes, but they maintain their nuclei, and they become like this big blob of nuclei that kind of shift around. But if it is unicellular and eukaryotic, and it's not plant, animal, or fungus, it is a protist. So while it's likely that protists share a common ancestor, the exclusion of other eukaryotes means that protists do not form a natural group or clade. This is a really diverse group, and we just kind of lump stuff in it. And you'll find when we get to plants in a couple of weeks, you're going to see some things that we're categorizing today as protists also then categorized as plants, because it just depends who's doing the categorizing, because people kind of have their own opinions about how that should be done. So protists are very diverse, which is what we were just saying. So our first eukaryotes, that's a cute little guy. I like him. So many species of protists are more closely related to plants, animals, or fungi, right? And we can kind of lump them that way because they tend to lean more towards one of these types of organisms than they do each other. So here are our main types that we've got. So we've got those that are similar to plants, and the green algae is going to be the big one here. Now, this is the one you're also going to see categorized as a plant. Give it a couple weeks, it'll show up again, and they're going to call it a plant. Because again, it really depends on who's doing the categorizing. So green algae are the closest relatives to land plants, and we think that land plants evolved from them about 475 million years ago. And some are going to include algae in the plant kingdom, which is what we were just saying. Now, algae are split into two groups, chlorophytes and charophytes. All right, and both groups contain both single-celled and multicellular algae. So your charophytes are the ones that are most closely related to land plants, and they're found in freshwater environments. And then your chlorophytes are marine and freshwater. So when you talk about the multicellular green algae, you're talking about stuff like seaweed, right? Seaweed can be huge, right? It's huge, huge, huge. So it's odd to categorize it as a protist and not a plant, right? But then when you start looking at it and you're like, well, it's really more similar to one of these or one of, it gets really kind of touchy in there. So there isn't like a right or wrong when it comes to that. Now, protozoa is what we looked at today. And these are the ones that are more animal-like. And you guys probably saw for the ones that were still alive, why they were more animal-like. They were moving, right? Especially the euglena. They move really fast. So the term protozoa is just sort of an informal term that lumps together the single-celled eukaryotes that are either free-living or parasitic. And these all feed on like organic debris. So if you go look at these guys, they're green. So how are they, where's their energy coming from? Yeah, photosynthesis, but these are autotrophs. These guys are heterotrophs, right? And they're heterotrophs in such a way that they feed on things. Now, when we get over here to the fungi-like ones, you'll find these guys absorb. So just like a fungus out in the environment will absorb its nutrients, an animal will eat generally, and plants will produce. That's kind of how these are lumped. So historically, your protozoans are regarded as your one-celled animals, but they're not really, because they possess animal-like behaviors, motility, which you saw, predation, and they lack a cell wall, which is found in plants and in algae. Now, the cell wall in algae and in plants is composed of the same substance. Do you remember what the cell wall is composed of in plants? Cellulose, right? Okay, keep that in mind. So historically, your protozoa are divided into four groups. You got your amoeba, and again, sometimes there's an O in this word, flagellates, ciliates, and sporozoa. Now, if we look over here at our fungus-like ones, these are the molds, okay? These are not mushrooms, these are molds, all right? And these guys absorb from decaying matter, which is what fungus does, right? So it makes sense that they lumped them over here. And they reproduce using spores the way that fungi do. So in other ways, they're quite different though, and they're more like protists. So for example, their cell walls are made of cellulose. So the same as plants, right? And the same as these green algae guys. But fungus, the cell wall of a fungus is composed of something called chitin. It's totally different. So they do have cell walls like plants, but they're composed of something different. So somewhere between evolving from this blob into a mushroom, we switched out what our cell walls were made of. So like other protists, they have complicated life cycles with both sexual and asexual reproduction. They're motile during some stages in their life cycle, and the two major types are slime molds and water molds. If you're looking to find a slime mold, if you've ever seen, because this is a common one around here, and you find it in houses or in public bathrooms, but if you've ever seen the pink stuff in a toilet, like the pink slime, that's a slime mold. There's your protist, your pink slime mold. That one's pretty common around here. So protists, ancestors, and descendants. So we have the idea here that there was a common ancestor, and then we branched into our protists over here. And they've got 6 major groups. And then we think our plants kind of branched off somewhere in here, and our fungi and our animals may not have come directly from these, but they probably had some common ancestor in between, is what we're thinking. So all eukaryotes are descended from early protists, but modern protists are very different from their ancestors. Like other eukaryotes, they've been evolving for the last 2.5 billion years. So protist structure and function, what's that in the picture? We all saw a good one today. Alex said it.
Paramecium.
It's a great paramecium. Yeah, Alex had a really good one. That was a really, really good one. So how do they move? So the three that we had today moved with these three different examples. So we have cilia, and you can think of them like if you've ever seen a crew team, the guys with the oars, right? And then pseudopod, and then flagellum. Okay, cilia is also another word used for hair. So you may hear about it when we get into anatomy. You may see the same word used again when referring to like nose hairs, ear hairs, parts of your small intestine that look almost like little hairs, little finger-like things that stick out and absorb. Can anybody break down the word pseudopod?
Fake.
Yeah, there you go, fake or false or
Fake, but-
similar like, yeah. Mhm. Yep. There you go. So pseudopod. And then does anybody know a similar word with flagellum? Like, have you heard that used before? Maybe as a verb. You ever heard of the term flagellate being used like to whip someone for punishment? Okay, there you go. It's a whip. Same thing. All right, so most protists are modal, but different types of protists have varied modes of movement. So protists such as euglena, which I think we all got to see a good euglenas today, have one or more flagella. That's going to be your plural, which rotate or whip.
Is it a brownie? Is it supposed to be mobile?
No, modal is actually,
What is-
yeah. It means that they're moving themselves as opposed to just being able to be moved. Fun one, huh?
Self-propelled.
Self-propelled. So they can whip like this, but they can also rotate. That's like a little motor on the end, right? And then you've also got your paramecia with their little rows of tiny cilia, and they beat and they swim. Now, if you're looking at it this way, it would appear that the thing is just got it like around the edges, but really it's completely covered, right? You're looking at a cross section. So it's completely covered with these little hairs, and they all just kind of wiggle around and they move. And as we saw, they were really effective at making very quick turns, right? And then other protists, such as amoeba, form cytoplasmic extensions called pseudopodia anywhere on the cell. Anchor the pseudopodia to a surface, and they pull themselves forward so they can kind of stretch out an arm sort of and squish forward. Now, these guys also use these pseudopodia to eat. It's very similar to some of our immune cells that use phagocytosis, where they'll take their little arms sort of, and they'll reach out and they'll grab whatever it is they want to eat, and then they'll combine it at the end and make a bubble and bring it inside, and they'll pop the bubble and they'll eat it.
You held something and it went into your.
Yeah, it's weird, isn't it? It's really cool. So some protists can move toward or away from a stimulus, and that's called taxis, when they move toward or away a stimulus. So protists can accomplish phototaxis, movement towards a light, by coupling their locomotion strategy with light sensing organs. So some of them do have organs that can sense light, sort of like our little flatworms. Do you remember our little platyhelminths? They could sense light, they move toward and away from it. They're way more evolved than these guys, but still. So how do they reproduce? Different ways, very different ways. So first one is what we'll refer to as binary fission, which is exactly what it sounds like. Fission means to split, binary means two. So you'd start with one that looks like this, and it would elongate and it would start to pinch off. And then what does this look like? Does this look familiar? We've seen this before. What does it remind you of?
Mitosis.
Yeah, it's pretty much mitosis. That's exactly what you got here. It's binary fission, but it's making a genetic clone of itself when it does this, right? All right, then you've also got slime molds, true slime molds. They'll exhibit what they call multiple fission, where it's just a simultaneous division into many daughter cells. And this is also how some of them get to the point where they just have like all these nuclei and like no real membranes, and they're this big blob of stuff. And then others will undergo budding.
Well, in that situation, what's considered an organism? Is that one organism or many?
See, that's the thing. They consider it they consider it like a colony of an organ. It's kind of like a hydra. You know what a hydra is? It's like multiple organisms that kind of form one organism. I'll have to find a hydra for you. They're interesting. And it really depends on who you're talking to, because this is going to be one of those. Yeah, got one of them. This is going to be one of those where different people are going to have different opinions on how to categorize it, because they're so amorphous at that point, but they all still have individual nuclei, and you don't
What's the
generally.
definition of organism?
Yeah, exactly. Yeah, exactly. So others will undergo budding. So this is when they make these little tiny buds on themselves and then they pop off and it's identical to the host. Yeast do this. If you've ever watched yeast under a microscope, they'll undergo budding. You'll get this little outcropping little pocket on it. It'll grow bigger and bigger and bigger. That'll just pop off like a little bubble. And that's how they make a new yeast cell. So fungus-like protists were once classified as fungi because they produce sporangia. So a sporangium is a structure that produces and contains spores for asexual reproduction. So when we talk about fungus in a little bit, and then also when we get into seedless vascular plants, we're talking a little bit about spores. But spores are, they're like sex cells in that it's one cell that then grows into a new organism, but they're not sex cells in that they are diploid always. So they don't contain half a genetic material. They just contain all the genetic material. And then for like ferns, for example, there's seedless vascular plants, it'll just drop to the ground and make a new fern from this one cell. So sexual reproduction with these guys involves meiosis and fertilization. And that's not uncommon. And some of them can actually switch whether they're going to use sexual or asexual reproduction based on usually environmental conditions. That is a handy thing to be able to do. There are actually some higher organisms that can do this. There are actually some reptiles that can do this in particular. There are some species of lizard that based on environmental conditions can choose to undergo a form of asexual reproduction called parthenogenesis, and it involves only female lizards. And it's a kind of a fascinating thing, but it's like you can get one female lizard can like trigger another female lizard to produce diploid. It's almost like diploid eggs. And then you produce clones of yourself at that point.
Yeah.
That's pretty cool.
And they would do that if there wasn't any males around, but if there's males around, they would want to sexually reproduce.
Correct.
Add more.
And it may also be a thing where maybe it's a resource related thing sometimes.
Yeah.
So conjugation, is another type of sexual reproduction that mainly only occurs in ciliates. And in this process, your nuclei from gametes come together and fuse and make what they call a zygotic nucleus. So sexual reproduction is often associated with periods when nutrients are depleted or environmental changes occur. Sexual reproduction may allow the protist to recombine genes and produce new variations of progeny that may be better suited to surviving in the new environment. So you can see why if you've got an environmental change and your species is struggling, it would make sense to be like, oh shoot, we need to shuffle things up a bit. Let's have some sexual reproduction for a bit here and get some new genetic combinations. So protist life cycles can range from simple to extremely elaborate. Certain parasitic protists have complicated life cycles and must infect different host species at different developmental stages to complete their life cycle. Some protists are unicellular in the haploid form and multicellular in the diploid form, a strategy employed by animals, right? That's what we do. And other protists have multicellular stages in both haploid and diploid forms. A strategy called alternation of generations that's also used in plants. So we'll be talking about alternation of generations in the next couple of weeks when we get into plants. So ecology of protists. I loved this picture. Look at that. That is so pretty. That's really cool. Sparkly. So autotrophic protists. All right, we start talking about our autotrophic protists. Yeah, I like them. So protists that perform photosynthesis are going to be autotrophic. What's autotrophic mean again?
Creates its own energy.
Creates its own, own food source and energy. So the position of photosynthetic protists at the base of the food chain makes life possible for lots of things. It says here aquatic diversity, but it's more than just that. I have a video in a minute that'll show you. So they feed fish, whales, support coral reefs, provide shelter to marine life in areas where sewage is dumped. They recycle waste. However, when the amount of waste is excessive, you can get these things called algae blooms. And then you get enormous masses of cells, of algae cells, and some of them can release things that are toxic to humans. And Like you'll hear about, oh, there's a red tide. Ooh, last night we heard a lot about tides, didn't we? There'll be a red tide or a crimson tide, right? And you don't want to go in the water because it's dangerous. So it's actually kind of a threatening name. It seems like a weird name, but it's kind of a threatening name, right? Because they're very dangerous. So what's the most important life on Earth? It's going to be these little photosynthetic protists that you probably never really even think
They
about.
create like 70% of oxygen.
Exactly. So as Michael just pointed out, these guys are what's responsible for most of the oxygen on Earth. And not just actively producing it, but the fact that there's oxygen here at all. So this video is a little bit longer, but it's really worth watching. And I want you to keep in mind when they start talking about like all of the things these guys do at the bottom of the food chain. And if you remember several years back, we had a terrible time when the rainforests were on fire and people were like panicking, and there were all these memes going around about how the Earth's lungs were gone. And that's not really accurate. I mean, it's not good that the rainforest burned, but the Earth's lungs are really the ocean due to these guys. All right, I'm gonna grab a snack while you guys watch this one, okay? This is a good one.
Thanks to the Monterey Aquarium for partnering with us.
Yeah,
Episode
yeah.
of SciShow. They hope you have a plankton of fun watching. Right now, there are incredible creatures in the ocean with the power to reshape the planet's atmosphere. After all, they've done it before, and you've probably never even seen them unless you've looked at seawater under microscope. I'm talking, of course, about phytoplankton, the microscopic critters that almost all life in the oceans and arguably on land depends on. Phytoplankton, a catch-all category for all the tiny aquatic organisms that can turn sunlight into food via photosynthesis. And there are all kinds of different organisms included in it, from cyanobacteria to whip-tailed dinoflagellates to armored diatoms, which look amazing under a microscope, by the way. Diatoms are essentially algae that build little houses out of glass or silica, to be precise, and they come in a stunning diversity of forms. In fact, if you've ever spread diatomaceous earth around your garden or your house, you were sprinkling fossilized diatoms. There are also coccolithophores, which can look kind of like a soccer ball covered in frisbees, thanks to their teeny tiny shells made of calcium carbonate. In fact, England's famous White Cliffs of Dover contain the fossilized remains of countless coccolithophores. And in today's ocean, the smelly compounds they produce help give this its unmistakable scent, a smell birds like albatrosses use to find their meals. The compounds that make those smells can also travel up high into the atmosphere to help clouds form and grow, making them more reflective and helping to keep us down here a lot cooler. And dinoflagellates are also very cool, don't want to leave them out. They have two tail-like flagella that help them swim around, and many can produce their own light, which makes for some really magical effects. But too many of them in one place can create a harmful algal bloom, a phenomenon you heard referred to as red tide, which is just one example of how these super teeny creatures can have a big impact on the rest of us, even us landlubbers. No matter what the phytoplankton take, they are unbelievably important to all life on Earth. First off, they're the foundation of the marine food web, underpinning everything from anchovies to whales. In fact, with a wee bit of help from seaweed and aquatic plants, they manage to support life in the ocean despite being outweighed by roughly 5-to-1 by organisms that don't make their own food. And if that's not enough, they are also atmosphere-altering machines. They consume about as much carbon dioxide as all of the world's forests and other plants combined, pulling roughly 11 gigatons of carbon from our atmosphere every year, which is especially impressive when you consider that by
Sorry, Ethan needed to come in.
mass, there's about 450 times as much plant life on land. Plus, phytoplankton account for more than half of the planet's yearly oxygen production. But even more importantly, they're kind of the reason we have oxygen in the atmosphere at all. Back about 2.5 billion years ago, near the boundary between the Archean and the Proterozoic eons, the Earth was a very different place. There were green seas and skies, and the atmosphere was likely a mix of gases like ammonia and methane that would immediately kill us. Then cyanobacteria evolved and ran rampant pumping out a huge amount of oxygen in what's now called the Great Oxidation Event. All that oxygen helped turn the ocean and skies blue and likely jump-started the evolution of complex life. And then around 540 million years ago, phytoplankton did it again, exploding in numbers and bringing us up to more or less the levels of oxygen in the atmosphere we have today. Paleontologists think the boom may have occurred thanks to nutrients weathered out of rocks by giant continent-sized glaciers. Or a more recent idea is that it happened because the plankton evolved a more efficient internal machinery related to photosynthesis. Either way, this set the stage for life to fully spread throughout the ocean and onto the land. Since then, they've calmed down on the reshaping the entire planet front and adopted more of a stabilizing role. For example, paleontologists think the evolution of calcifying plankton balanced ocean and helped protect the seas from mass extinctions. Before about 80 million years ago, the ocean was not a super stable place to be. Changes in the chemistry of the atmosphere from events like volcanic eruptions often caused deadly swings in seawater pH. Then some lineages of plankton, including those ball-like coccolithophores, figured out how to make shell calcium carbonate. Soon, they took over the open ocean. And over a relatively short period of time, so many of their shells sank to the depths of the ocean that they created a thick layer of calcium carbonate ooze on the seafloor. That ooze acts as a chemical buffer in the ocean, a natural braking system on runaway acidity. See, the shells are solid at a more neutral pH, but as the water gets more acidic, they start to dissolve, releasing carbonate ions. Those chemically react with the surrounding seawater to push an acidic pH back towards neutral. Now, there had been a minor version of this in the shallows, thanks to mollusks and other large-shelled organisms, but it took phytoplankton to make it ocean-wide. And the end result was a more stable ocean chemistry, which meant marine life became less vulnerable to things like volcanic eruptions. Even today, this chalk layer is helping buffer the acidifying effects of all the carbon dioxide that we humans pumping into the atmosphere, though there's only so much it can do. When we consider everything lives in the ocean, from majestic whales to bountiful coral reefs, these little unassuming are easy to overlook. But if you look at the history of Earth, it doesn't take long to realize that though they are small, they are also mighty. And they deserve as much fascination, love, and protection as any other living thing on this planet. Thank you again to the Monterey Bay Aquarium for partnering with us on this episode.
So little, like you said, small but mighty. I mean, for real, none of us would be here without those dudes and similar organisms. So heterotrophic protists. All right, so some heterotrophic protists engulf and digest their food. Others live while absorbing molecules from their environment. So the engulfing kind would be like an amoeba is a great example, right? So amoebas capture and digest their food surrounding a cellular particle and then taking it inside themselves to form a food vacuole. A food vacuole is a small cavity in the cytoplasm that temporarily stores the food. So they'll reach out, grab it, bring it in, hang on to it till they're ready for it. Paramecia and other ciliates use their cilia to sweep food particles into their gullet, an indentation on one side of the organism. So that would be like right here. And they use all those little hairs, sweep, 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 sweep. And they sweep it into, it's like sweeping it right into their little gullet, and it brings that inside of them. And then they can use that to get some energy from. And then slime molds and water molds are important recyclers of organic material. At one stage of their life cycle, some slime molds fuse to form, like we were saying, large cells with many nuclei. And these structures are known as plasmodia. So Michael, when your question, like, is it one or is it more than one? They're just gonna call it something completely different. So as not to answer the question, they're gonna call it a plasmodium, or plasmodia is plural. So your sporangia develop from these plasmodium. So some of these are symbiotic critters. So some protists have symbiotic relationships with other organisms. So here's one, Trichonympha. That's a cool looking guy. It looks kind of, I had something that looks similar to that in one of my slides today. Has a mutualistic relationship with termites. So this is the one that lives in termite digestive systems and helps them digest wood. Because remember, plant cells have cell walls of cellulose. We can't digest cellulose, neither can termites, and all they do is eat wood. So it's actually these guys that live inside of termite digestive systems that make it possible for them to keep a diet of wood. Other protists are parasitic and can cause diseases. So there's one. Trypanosoma causes African sleeping sickness. You've ever heard of that? Not a good thing to get. And then we've got the Protist plasmodium, is what they're going to call like the sort of this general one. This can cause malaria. Now, where do you get malaria from?
Mosquito.
Mosquitoes, right? So this is one that has a host and moves from host to host, right? It's this dude here. And then we go on to fungi. All right, fun stuff. These are great. I love this picture. I found this picture online. I was like, I have got to use that. That is stunning. So fungi are eukaryotic heterotrophs that have cell walls, and the cell walls contain chitin. So remember that cell walls of fungi are not composed of cellulose, but rather chitin instead. So most of them are composed of these thin filaments called hyphae. All right, so when you see a mushroom outside, all you're seeing is what's called the fruiting body. It's the sexual organs, the sexual reproductive parts of that fungus. Most of it is underground and you can't see it. And they have these hyphae, which are like hairs, little tiny hair roots, and they spread everywhere. And that mass is called a mycelium. So what you see above ground is the reproductive structure, and the hyphae are underground, and they form, quit spam risking me, this mycelium. So most fungi reproduce both sexually and asexually. Asexual reproduction can occur when cells or hyphae break off and begin to grow on their own. And some also reproduce asexually by means of spores. So if you have any mushrooms at home, baby bellas, portobellas, buttons, whatever, anything, especially the perfectly round ones are the easiest ones to do this with. If you have any at home, get a piece of white paper, take the stem off, turn it over so that it's cap up, so that these gills are hanging over the paper, right? This is where the spores are stored. If you've ever touched one and come back with like a brownish black dust on your fingers, that's the spores. If you leave it there overnight and come back the next day, pull it up, it will leave a print like a stamp. And it'll show you exactly where all the little spores fell. It's really pretty. It's almost like all fireworks or flowers or something. It's kind of a fun thing to do. So most fungi can reproduce sexually. Spores are produced in structures called sporangia. And some of these fungi are going to have like a minus and a plus type. It's sort of like a male and female, but not quite. They'll call them a minus and a plus, and they'll fuse their nuclei together that way. So these are super ecologically important. Look at that gorgeous thing. They're so pretty. So they don't ingest their food, they absorb it, right? So remember when we talked about ecology and how fungus was important at bringing nutrients back into the soil, back into the ground, right? They help maintain your equilibrium in nearly every ecosystem because they recycle the nutrients. Some of them are parasitic and they cause serious animal and plant diseases. So if you've ever had athlete's foot or thrush, babies with thrush or any kind of yeast infection, that is a fungus infection you can get. Yeah, athlete's foot is a fungus in your feet. Nail fungus gets in your feet. Babies can get thrush really easy. They get yeast in their mouths. It can grow in their mouths. And these things they're common fungal infections. And of course, you've probably seen some trees that get fungal infections. Sometimes that like warps the leaves, or they can look really weird looking, or it can get into the bark almost and start to break it down and then damage the tree. So some are mutualistic, in which both partners are going to benefit. And then you got lichens. So lichens are symbiotic associations between a fungus and a photosynthetic organism. So the photosynthetic organism provides the energy and the fungus provides the water. So everybody knows what a lichen is? You ever seen it on a tree? They're kind of feathery looking. They look like moss, but not really because they're flat and then papery. So it's a combination of a fungus and typically like a blue-green algae sort of a thing, like a cyanobacteria type thing, or just a green algae. And they'll combine together and they form this new symbiotic relationship that looks like a completely different organism. So mutualistic associations of plant roots and fungi are called mycorrhizae, and the plant roots are woven into partnership with the web of fungal hyphae. Now, this is a fascinating thing that we are just starting to understand, like kind of just starting fairly recently to understand it. But it's the fungus that help the trees communicate. Basically, mycelium in a forest floor will build a telecommunications network between surrounding trees through the roots, which is pretty fascinating because it helps the whole forest communicate and stay healthy. It seems almost unreal, but this shows you how it works, and it's pretty cool. Hopefully this one isn't as bumpy as the last one.
The idea of talking trees has been capturing the human imagination for generations.
Go away.
It's worse.
Okay, so maybe they don't talk to us, but it turns out trees can talk to each other.
Speaking to each other.
But that does beg the question, what do trees have to talk about? And can we learn to speak their language? Underneath the soil, a vast and interconnected network of life links the trees through their root systems. But they can't talk to each other without help. The whole process starts with hub trees, the oldest and tallest trees in the forest. Hub trees have greater access to sunlight, and through the process of photosynthesis, end up producing more sugar than they actually need. Underground, fungi need sugar to survive. Most of their bodies are made up of a mass of threads called mycelium. They grow within the root system of trees to absorb the excess sugar. In return, the mycelium provides the tree with the nutrients it needs from the soil. This symbiotic relationship is known as mycorrhiza, which stems from the Greek words for fungus and root. These tree-fungi relationships connect the trees in the forest together, forming an underground communication network to exchange water and nutrients, nurture their seedlings, and send warning signals when under threat. So, how many trees are really talking to each other? To get a better picture of these forest relationships, a team of researchers used DNA analysis to map a fungal network in a patch of Canadian forest. Remarkably, they found that one tree was connected to 47 other trees. Their models also showed that when hub trees were removed, it would cause more connections to be lost than if trees were simply removed randomly. Studying these kinds of underground exchanges will play a vital role in creating stronger, more resilient forests for the future. Even though we might not be able to talk to trees, at least we can still keep trying to understand their language. Who knows what they might say?
So when we did our ecology unit and we talked about deforestation, right? So there's a lot of, remember you can just clear cut, just blow through stuff, or you can go through the whole process of selectively removing trees. If we knew which trees were hub trees, we could be very careful about what we selectively remove, and it can keep the entire forest healthier, which would be ideal, right? And so it may be as basic as just kind of doing enough research to come up with a general rule that it's typically, like they said, the oldest and tallest. And if we find a way to leave the oldest and tallest, maybe we can keep everybody healthier. In my old house, our old house had a forest behind it that they knocked down to build a subdivision. And yeah, and there was like a sort of a gully or low point in the forest. And at the bottom of it was this massive, massive, massive chestnut oak. I mean, massive. It was probably as the diameter was at least that of the table. It was huge. And it was so big and everything ran down to it because it was in a depression. So it got lots of nutrients. And I was thinking, well, that one's so fabulous. They're going to leave it when they build this neighborhood. And they took it down. I mean, 3 of us could stand on the stump. It was so huge and it was so sad. But it makes me wonder, when you remove something like that, how does it impact everything else around it? Yeah. And then, like I said, they're just kind of learning about this sort of stuff. This is really new, cutting edge business. So some fungus is actually bioluminescent. So you've got fungus that can do its thing and glow, right? So these guys are doing neat research too.
Right now, we're not sure why the fungi bioluminesce. And that's what we're trying to do right now. Let's figure it out why. So organisms bioluminesce for various reasons, but it's probably split out from the basic detoxifying processes that happen in the cells. And some of the antioxidants, as they break, give off light. As these traits of other organisms go on to gain additional functions, like fireflies use their light system for mating and communication between species. A lot of that also happens inside the marine environment. But right now, we're not exactly clear as to why mushrooms.
The experiment that I'm performing out in the field is an ecology experiment where we are trying to understand the interaction of the mushrooms with insects. And so to do this, we're using fake acrylic mushrooms that we've produced in the lab and putting them out in the field covered in glue. Some of them have an LED light that has about the same wavelength as the so it's producing the same color light. And so we're comparing the amount and the type of insects that are on the dark traps to the light traps. And by looking at the 2 sets of data, we can tease apart, which insects are attracted to the light and which insects are repelled by the light and which insects just don't care about the light. Through this, we can better understand the interaction of these mushrooms with their environment.
It's very likely that the bioluminescence from mushrooms has some ecological significance in the forest. Based off of our early results or early data, it definitely looks like insects are attracted to it. What role those insects are doing requires further study. Fungi are at this special moment right now where the research is really beginning to progress, and we're likely to figure out a mechanism within the next few years.
They actually have some, you can buy some that will grow here. I think they're orange though, not green. So looking over our notes. So your protists are going to be any organism, plant or animal, fungus or prokaryote. Any organism is not a plant or animal, fungus or prokaryote. And then why are brown algae considered protists even though they're multicellular? It's because they're going to be more related to some unicellular protists than any other kingdom. Why do scientists no longer use categories like ant-like, plant-like, and fungus-like? It's because they don't really show the evolutionary history. And we are always in biology moving more towards showing evolutionary patterns than we are just physical similarities, right? So most single-celled eukaryotes are going to be classified as protists. The genetic analysis indicate they belong to 6 different clades. They were the first eukaryotes, and unlike protists, which are unicellular, kelp has differentiated tissue. So this is one of the multicellular protists that we talked about. This picture here, these are diatoms. So when I say I love them because they're beautiful, that's what I mean. That is so cool. And they're all different. There's some really beautiful, there are some people who will take, you can find it on Etsy, for example, but they take high-powered, high definition microscope pictures of these, but they arrange them like a kaleidoscope image or almost like a mandala. And they'll take pictures for artwork, and some of them are absolutely unreal. I mean, the amount of time it would take to move these little microscopic organisms perfectly into place and create this image, they're beautiful. Yeah, it is. Well, there's actually lots of microscopic art. There's a guy who does sculptures on the head of a pin. Like, yeah.
Or just like, I do.
I saw Godzilla carved out of the green rice.
Out of grain of rice.
Godzilla.
Yeah, that is cool.
The smallest Godzilla to ever exist.
There you go.
I saw one where a guy took a pencil and he carved, I forget what it was, he carved the end of it or something.
Oh, wow.
It was very.
Wow.
You can probably do that.
So our trues and falses, the first eukaryotes were archaea. No, they're going to be protists. Is it possible to find the earliest fungi by looking at modern protists? No, not really. Modern protists have evolved very little from ancestral forms. No, they're still evolving, right? Today, there are 300,000 known species of protists that we know, probably a whole lot more that we don't know. I'm sure there are some down in the depths of the ocean we have yet to even locate, you know. The oldest known eukaryotic fossils are about 1.5 billion years old, and protists evolved way longer than 3.6 billion years ago. It was more like 5.7. Plants, animals, blah, blah, blah. Okay. And then what are pseudopods? We talked about them before. They're false feet, temporary projections of cytoplasm used for movement. How do amoebas capture and ingest food? They surround the food particle and ingest it, which we talked about. And then I'm sure you guys could have drawn these, but I like these dudes at the bottom too. Pretty things. They're so pretty. So here's your little analysis when I talked, little analogy, sorry, when I talked about the paramecium being almost like a crew team. That's a great example. And then in this one, they've got the little oar going back and forth, right? And you could also think of like how a motorboat spins, right? They can also spin. So when they reproduce asexually, sorry, by means of mitosis, remember we typically call that binary fission, right? And then we talked about that, and we talked about that, and that. So on to this last bit, your autotrophic protists. These guys, remember, you're going to be the base of the food chain, responsible for the pH of the ocean, responsible for the fact that we have oxygen in the atmosphere. Always keep them in mind. Phytoplankton are the most important organisms on the planet as far as sustaining life goes. And then over here, this is a great diagram here of this paramecium. So if you had worked on this in your notes, you're probably pretty familiar with what you were looking for when it showed up under your microscope today. And if you didn't get it, Alex had that really, really good one. Let's see. Here's our symbiotic one. So we talked about termites and their inability to eat wood, right? And that was here in your notes. Now here's your plasmodium stuff. And this shows kind of how it works here. So you've got this mosquito bites a human, and then these plasmodium sporozoites enter and they infect liver cells, and then they turn into a different type of cells, and then those infect blood cells, and then those burst open and all the little gametophyte spore type things are released, and then another mosquito bites that person and sucks up the blood and gets those in it and then squirts it into the next person. Because mosquitoes, when they bite you, they inject a little first so that they keep your blood flowing. They like ticks do too. They inject a little bit of chemical into you that keeps your blood from clotting so quickly so they can just suck more out. Isn't that nice of them? Yeah, that's why it itches so bad. So slime molds, remember, they're going to be heterotrophic, and we already went over that. We went over that, and we went over that. I like that picture too. So they're responsible, remember, for breaking down pretty much all your organic matter. If you didn't, there was a question about it when we did our ecology lab outdoors, where it said something like, what would happen if there was no fungus? And most of y'all were like, well, nothing would get broken down. There'd just be piles and piles of stuff, which is true. No decomposers, but also you wouldn't get any of the nutrients returned to the soil, which is responsible for building up new life. So you'd be missing that too. Does anybody know what a fairy ring is?
I do. Is it a ring of mushrooms?
Do you know why they go that way?
Because there's nutrients near this in your food.
The nutrients there, yeah.
It's all the same organism.
It's all the same organism. So they're all connected underneath. All those, the mycelium underneath, yeah, is all connected. And they're they'll produce one, and then they'll produce another, and then they'll sort of, all that mycelium goes in all different directions like a web. And they find themselves in this nice little circle. And if they're little toadstools, then people look at them and go, oh, look, the little fairies had a meeting last night. It's a fairy ring. Ta-da, right? And yeah, there can be something in that area that they're feeding on that's obviously going to be of interest to them, or they wouldn't be growing there. So you guys, I'm going to go over something with you guys that I'm just,
circle so it can be equidistant, right?
yeah, that's part of it. I'm going to share something with you guys that is going to be in your email this week, but I want to, we have a few extra minutes, so I want to go over it with y'all. All right, so
We have a lot of time.
there's some, yeah, we do. There's some weirdness going on.
Some weirdness.
There's some weirdness going on, and I'm going to give you guys a heads up on it before we get, yes, so thoracic spine. All right. So the reason this is in your email this week is because we may end up, are we listening? Are we listening, guys? Cool, Michael. We may end up having to make some scheduling changes in the few weeks ahead, which I'm hoping will not be the case. But I've been having pain for about 6 weeks, and my doctor and I thought, okay, it's just lupus related pain probably, because it's definitely nerve pain. So we did all the things we're supposed to do multiple times and it didn't fix it. So he says, go get an MRI just to be sure that it's not anything structural. It's something structural. I actually have ruptured a disc in my thoracic spine. So your thoracic spine is this part of your spine, the part that's through your rib cage, through your thorax, the center of your body. And your cervical is up here. And because a lot of my pain was in my neck area, the MRI only covered my cervical and like just the top of my thoracic. So I don't know what else is going on down here, but I have a sneaky suspicion I'm going to end up with another MRI to find that out. My issue is right here at between T2 and T3. So these are all numbered. All the cervical ones are C1, C2, C3. And these, you'll see them as either T or TH. Sometimes they'll do them as TH. But my issue is right here at T2, between T2 and T3. Now, what happens here is, well, first, let me show you kind of how this works. So you have discs. We haven't done this genetically yet. I mean, anatomically yet. We're getting there. So you have these discs in your spine, all right? And they're located between each of your vertebrae like this. All right, and this is what we would call a lateral view. And we're looking at it from, it used to be like standing on a person's left if you could cut them in half right down the center this way, okay? So you've got these in the middle. So between each vertebral body is a cushion called an intervertebral disc. And the disc absorbs stress and shock that the body incurs during movement, and it prevents the vertebrae from grinding. So when you twist and turn, right, that's all because these guys are nice and slippy in there. So they are the largest structures in the human body that don't have a blood supply. Okay, they're not vascularized at all. But what they have is this jelly-like center. It's a squishy, almost like a sticky jelly. No, it's not cartilage. It's a different material. It's called nucleus pulposus, but it's this mushy jelly-like material. And then it's wrapped in this material called an annulus. It's annular fibers, and they're really tight. You can think of it almost like Kevlar, right? And it wraps around it and it keeps it all enclosed, all right? So your spinal cord is going to run from your brain, right, all the way down to the bottom of your spine. And it runs through a hollow tube because each of your vertebrae is going to be hollow, right? So it's going to run all the way through, all the way down. And then these guys are going to sit sort of next to it. I'll show you a picture in a minute where you can see it. And they keep everything from grinding around, right? And then there's some joints and stuff in there that move. So there's an opening on the left and the right of each vertebra. You can see like right here. And then you've got nerve roots that branch out from each one. So the nerves that come out of each one goes to the left, one goes to the right. And then they further branch out like trees, right? And they go everywhere, all of your organs, all of your muscles, your skin, everything. But they start in these places. So when an intervertebral disc gets ruptured, it means that that annulus is torn and the nucleus pulposus inside, that jelly liquid, jelly substance, will ooze out. And it can cause it to ooze into a space where it's not supposed to be, which means it can press on one of these nerve roots. And then the other problem is that it never comes in contact, or it's not supposed to come in contact with anything else in your body. So when it erupts or whatever, explodes and oozes into your body goes, yeah, your immune system goes, what's this? And it reacts. So your immune system attacks it. And when your body attacks its own tissues, it's called autoimmune, right? So what you end up with is a situation that looks like this. So this is in particular what I'm looking at. So you've got a healthy disc here. So you got your spinal cord that runs through here, right? In this little hole here. And then you've got your nerve root, like I said, coming out these two different directions. So this is a top down. This would be person's back and the person's front. So ventral, dorsal.
Those are the bumps.
These are the bump. Yes, these are the spinal processes that you can feel if you run your hand down your back. Correct. All right. And then they're joints here called facet joints. And one vertebra sits inside another. So this is how it's supposed to look. All right. This is what mine did. And it exploded here and it oozed out. And this is left paramedian. So you've got a ventral side of your body, which is the front. You got dorsal up here, and then you've got lateral left, lateral right. So if you're looking at this person, this is lateral left because this is their front, right? So your right is their front. So this would be my left. So it's oozed into this space here. Now, had it oozed this direction, easier to fix. It's oozed this direction, more difficult to fix. Plus, if you look back at where this joint is, this is T2 and this is T3, so it's right there. When they're down here, they're much easier to get to, as you can imagine, because you're not dealing with all of these other structures, right? So it's down here and it's squashing this nerve, okay? And this nerve root is, when my doctors, when we talked about it, he's like, well, that's a nerve dispersion pattern, what you're feeling. So it makes sense that, you know, that it's nerve involvement. So what is it? This is where I'm feeling it, but I'm feeling it only left-sided because only my left side is impacted. But all of this area that is in red is what hurts, which is not what you would think. You would think it would hurt right there, right? But it doesn't. It hurts everywhere the nerve goes. So when you get down in here into your thoracic spine, you find that these nerves control other things besides just muscles and whatever, like, oh, let's see, your heart, your lungs, those sort of things are kind of important, you know? So you don't want them to be hugely damaged. This is why if you break your back in a certain place, your heart might stop beating or whatever, because it can cut off that nerve entirely. So mine's being somewhat compressed, and what they said was somewhat effaced, which means the little tube here that the nerves are all wrapped in a fiber, it's thinning. The fiber's thinning because it's being pressed. So that's not good. So if you've got it going one side or the other, like I do, you've often got what they're going to call unilateral pain versus bilateral pain. Bilateral is both sides of your body hurt at the same time, which is typical lupus stuff. So when it was one-sided, we knew it was something different, but we really didn't expect it to be a structural thing. So it was kind of a big surprise. We were like, oh my goodness. So anywho, it's this whole area only on my left, and it is excruciating. I'm not kidding, like unbelievably excruciating. So what I've done is bed rest and oral steroids and pain meds, and I've done all that for a couple weeks because we've been trying to figure out what it is. But before, like right before I found out that it was a vertebral issue and not just like nerve irritation, I kept trying to wean myself off the steroids and stuff, and it got really bad. One day it was so bad I thought I was gonna throw up. It hurt so bad. It was really, really bad. So I'm going next Thursday, to a spinal specialist who's what they call a pain interventionist. And he's going to try to find some ways to work around it. But my doctor was like, and it was funny because ruptured discs are not uncommon, but rupturing that disc, where it ruptured, the fact that there wasn't an accident or anything to cause it, and the fact that I'm 45, which is old but not that old, is super weird. Super weird. So he's like, okay, I want to be on the phone when you're there. So we're going to have him on the phone while I'm with this visit so we can come up with some ideas. The likely first stop will be steroid injections into the disc, where they go with a big needle into your spine, jam it down into that disc, fill it up full of cortisone or whatever. And hopefully if you can get all the swelling that's around it from the autoimmune reaction and everything else to go away, it might slip back in and you might be okay. The other option is surgery, and they'll go in. There's multiple ways they can do this. So look back at this. So there are multiple ways they go about doing this. If they come in, and I've been researching the heck out of it because I don't want to go in there and not know what questions to ask, right? I like to know what questions to ask. So I'm going to ask this dude though, for real, anytime you can get a picture, I want a picture because I want to bring it back to my kids and show them gross things, right? So the old school way to deal with this problem right up in here, or any of these thoracic ones, if it's lateral, so on a side, right, is they go in what they call the intercostal space. So they go in over here on your side through your ribs, or they go in through the front. And if they do that, they make like a big incision and they go in through the front and they push all your organs out of the way and come to the back. And then they remove a rib, like take out a rib, goodbye rib, because it's in the way of what they're trying to do, because they're trying to get into this little space, like right in here, and the ribs all connect through here. So they just take it out. These are kind of hard to take out, these ones up tippy top, as you can imagine. So I did find a guy in Florida who has a new endoscopic laser thing. And I watched his endoscopic laser surgery because he will actually live stream them. And he's like, if I mess up, everybody's going to see it. And there are all these other like orthopedic spine surgeons and whatever neurosurgeons that are on the live stream asking questions, typing questions, or just asking them. It's almost like a Zoom and the dude's in surgery. It's kind of cool. But he had one right up in, it wasn't 2-3, it was 5-6 in the one I saw. So like right about here. And he went in with a 4 millimeter diameter tube, which is not very big, right? A tube. And he went in at, yeah, like a pencil, smaller than a pencil. Yeah. Mm-hmm. And he went in at a 45-degree angle, and he actually used a laser and an endoscope, and he used a dye first, and apparently the dye shows any degenerated parts of the disc or anything that's not healthy. And then if it shows up blue, the dye got it, so you know that it's got to go. And if it shows up yellow, it's calcified, which is also not good. And then he just sat there and just zapped this whole thing until it was gone, and then pulled back out and it was done. And it was like, and the guy after surgery could stand and walk, and I mean, like that. Because the old stuff, they said 4 to 5 days in the hospital, 4 to 6 weeks recovery, physical therapy, all that kind of stuff. So this newer stuff, they're getting really good. Huh?
What did he shoot with the laser?
The disc. Mm-hmm. And just like basically vaporized or atomized it, just took away all the unhealthy parts. It was really cool.
Doesn't that mean that there's not going to be rubbing there? There's no water.
Right. So they do. Yeah. So sometimes what they'll do is they'll fuse, where they will actually, like, it's almost like you put a bracket here and here around it.
Make them one.
They function as one instead of two, so you don't need the shock absorber anymore. So sometimes they'll do that.
Your back is slightly less flexible.
Yes, right. So they'll do that. And then the other thing they can do is I've seen some stuff. I don't know if it's gotten to people yet. You listening, Cole? I don't know if it's gotten to people yet, but I know they've done it with animals where they have like an artificial disc filler, basically.
Yeah.
So they'll suck all the bad stuff out, they'll eject the good stuff in, and then they plug it where the annulus is torn. And they basically build you a new, like a prosthetic disc within your back, which is really kind of cool. So these are all things. Yeah, they also found I had a lot of degenerative discs, which means in general, a lot of these discs from like here down were just not healthy, which I already knew. I already knew that. That's been something I've dealt with for years, but the ones I knew about were down here. I didn't know I had any up here. So I do have some up here. And the last thing they found was, I don't have a graphic for this because they didn't think it was as big a deal. At least my doctor didn't think it was. But they have these things called Tarlov cysts. I'll show you a picture because they're weird. But and I have no idea how I got these. I don't have an idea how I got any of this, except that it's more common in degenerative discs. So if you look like, okay, here's a good example. Here's a picture. So every nerve, your spinal cord and all the nerve roots that leave it are all wrapped in this dura. It's like the meninges that cover your brain. It's like packing tape, right? Basically, it keeps everything together. It's cling wrap. But if you imagine like a hose with a weak spot in it, if you shoot fluid into it under pressure, you're going to get a bubble, right? That comes out in a spot. And that's what these are. And they found probably 7 of them just in my neck. So they can press on things, but they don't know if that's what's causing any of the problems yet. This other guy, this neurologist guy, the neurosurgeon dude, whatever, he'll be able to be more specific about that than my doctor is.
What fluid is it?
It's cerebrospinal fluid. And so what I saw about those is sometimes they try and just drain them, but of course they're just going to fill back up. So I saw a thing where they drain them and then wrap them. So they drain it and then they basically wrap the nerve like you would wrap flex tape around a leaking hose is exactly what it is. So anyway, this is all weird stuff that I didn't know to expect and was really shocked by. And so was my doctor, because we expected it to come out totally fine. And we'd go, okay, it's just lupus crap. What do we expect? More anti-inflammatories, more steroids. It'll end sooner or later. But this isn't something I can ignore. Like I can't ignore the thing going on here. This is a big old deal. So we have to fix this somehow. So they're probably going to try stabbing it first, which does not sound fun to me. And then if that doesn't work, they'll probably do some kind of a surgery. So if they do some kind of a surgery, or even if they end up stabbing it, it may be the kind of thing where we have to go remote for a couple days. I am working to get as many of your PowerPoints and stuff done way ahead, because I don't want it to be something where I'm scrambling. I don't know what will be impacted. I know that if they do the injection, what to expect is feeling great for several hours, because they'll give you a local first. So then all your pain goes away and you're like brand new, and then it wears off and then it hurts like all get out for a while, like really bad. And then it starts to just decline. And if it's going to work, it just declines and goes away. And if that's not the case, then and I don't know otherwise what to expect. So anyway, I included this in the email so parents can see it. And there wasn't time to talk about it with the other group, but I wanted y'all to know. And as always, if you have questions, you can just ask me. Like I'm not afraid to answer questions about it. I don't know anything about what's going on more than y'all do right now, because it's all kind of weird. But I don't have any reason to think that it's gonna cause me any major permanent damage, as long as we can get it fixed. But like anything, if you're dealing with spine and nerves and stuff, those are risky things, because one wrong move and so yeah, I know that they do the injection under a local, which I kind of laughed at when I saw that video about it. So I told Jason, I was like, you're going to this appointment with me because I can handle just about anything, but the orthopedic stuff grosses me out. Like it really does. I don't like the bone saws and the needles and spines and things. Those things wig me out a little bit. Yeah. So if they're gonna stick something in my back, I'm gonna have to be sedated. So, and apparently you can get awful headaches during all this too, because they're messing with cerebrospinal fluid and it's all the same system. And like if you get the steroid injection, you can get like a 2-day headache that'll just knock you on your butt. So anywho, but I'm gonna share this with this week's email, and I'll probably put some links in it in case you wanted to look up some stuff. It should be pretty interesting. And then lastly, I am gonna make an extra credit assignment for you guys for this Omicron variant, and doing some research on what's coming out and coming up with some potential hypotheses as to what we could expect and then how we could test them, right? Because there are some things we can definitely
Test hypothesis.
Yeah, how would you test it? Like
How do you get sick from this?
if you were researching it, how would you test it? Like what would you be researching? Because it's in no, that's cysts. I already told you that. That's sad because those are very tiny. Sorry, did I say that out loud? I did. If you go to our Class Connect, I put in did anybody see the article that I included a bit ago? Okay, so the article here that I put in the forum.
Oh, I looked at half a second.
Yeah, so it's worth reading, and I've been adding other articles to the forum, and if you guys find something, you can add it to the forum as well, because it's pretty interesting. It takes me a second to get in here.
You should get that.
I have it, but I have it on my phone. Hello. So I have it right under here in this section here, where it's all bio. And this is the article I shared with you guys on December 16th. So like right the day after you left, I was like, oh, this is interesting. So I gave you all some background here on viruses, how they function, because we hadn't covered that yet before y'all left. And that the initial data was showing that Omicron is spreading more quickly. And they're showing that it multiplies faster in certain tissues rather than other tissues. They're finding in particular that it's not targeting lung tissue, or it's not as efficient at replicating in lung tissue as wild type and Delta. Wild type is your first, that's what you call it, wild type. So that is why we're seeing less people on ventilators. But they are also finding that it targets upper respiratory tissues more efficiently. So, and it seems to, the first place it seems to show up is tissues in the throat. Then it moves to the sinuses and nose. So we've been testing with nasal swabs this whole time. And now if you have an Omicron variant, it's possible that you could have it, test your nose and not see it because it hasn't migrated yet to that part of your body. It's already down here. So what they're doing now is they're trying to get some FDA approval for at-home throat swabs, which people are like, oh no, people can't do that. They're not as good at it, blah, 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 blah. So we'll see what happens there. But that's, yeah, throat swabs are gross. Like when you go for strep and it's like, bleh. But if it lets you know. So a lot of people are testing, and then they also, the CDC decreased that window to 5 days, right?
Yeah.
So when all that happened at around the same time, and right at Christmas, which was interesting to me that they did it right over the holidays and they did all this, because what we see now is like everybody has it, or everybody's been exposed. Like I know 4 times that I was exposed over the Christmas holidays, and all 4 times I was outside of the 5-day window before I even knew I'd been exposed like that.
Yeah.
So, I mean, that's what keeps happening now. So we're all kind of in that system. So my thoughts on this, when I gave this whole explanation, it's probably worth reading through because there's a lot of good information in there. So I'm interested in your hypotheses, but so mine were things like, for example, and you may have, yours may be the same as mine or different, but if this Omicron variant spreads quickly from host to host, but is less likely to cause severe or deadly effects to a host, then what? Like an if then. If naturally acquired or vaccine acquired antibodies are similar enough to lock down all or some of Omicron's identifying proteins, then what? And then if naturally acquired Omicron specific human antibodies or future vaccine acquired Omicron specific antibodies are able to successfully lock down wild type and Delta variant, then what? Of all the variants that have come out so far, this one looks like it may have potential to bring herd immunity. But that means that enough people have to get some level of immunity. And the question is, do they need it to Omicron or to Delta and wild type and Omicron, or just Delta, or just like which is going to apply? And we don't know yet, but it's super interesting. This may be the one. I have some hope. Yeah. So anyway, I shared that. I shared 2 other articles in here, and I found something else yesterday that I need to share. But I'm going to keep posting it, and then I'll add a little something to one of the next coming emails about how you can get some extra credit for it. But it'll be something along the lines of forming one hypothesis about Omicron in particular, and then how you would go about getting that data and applying it, and what it could mean, like what your potential outcomes might be, what you can infer based on what you know. Okay, so y'all are good. You got about 11 minutes before Jackie. Thank you guys. I will say goodbye to these folks here. Somebody said something.
By fake virtual people.
Yeah, bye virtual people. We don't even see it anymore.
One day you'll have a real battle.
One day to be here. Bye. Shannon says bye.